गुड आफ्टरनून एवरीवन। थैंक्स टू WNO। हम थोड़ा हिंदी में बात करता हूँ लेकिन इंग्लिश कंफर्टेबल नहीं है। मैं एक्सपीरियंस पोमोग्रेनेट फार्मर हूँ। आई एम नॉट ए कंसल्टेंट एंड आई एम नॉट ए साइंटिस्ट। ये यस सर। यस 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 सर। ओके। अभी हम अनार का फार्मर हूँ। मैं कर्नाटक से आया हूँ। कर्नाटक का चित्रदुर्ग डिस्ट्रिक्ट में हम 20 एकड़ अनार करता हूँ। उसमें 15 साल से हम अनार कल्टीवेशन कर रहा हूँ। अनार में इतना प्रॉब्लम्स है इंसेक्ट का और सम सॉइल बर्न फंगस का। कॉलर रॉट का ज़्यादा डैमेजेस था पहला, अभी विल्ट का ज़्यादा डैमेजेस है। रूट नेमाटोड है और वाइट ग्रब्स का डैमेजेस बहुत था। इसलिए हमने क्या किया, वो 3-4 साल हम काली केमिकल ही यूज़ किया। वो केमिकल यूज़ करने के बाद में वो 3-4 दिन कंट्रोल रहता था, बाद में फिर वो इंसेक्ट डैमेज करता था हमारा रूट को। अभी हमने नीम शुरू किया। फिर 300 ppm का नीम हम 2 ml 1 लीटर पानी में मिक्स करके 5 लीटर सोल्यूशन वो हम रूट के नीचे दिया है। वो अच्छा रिजल्ट मिला हमको। सो अभी ऐसे ही मैं 3 साल 4 साल 5 साल कंटिन्यूली हम वो नीम का
तो
इस्तेमाल हो रहा है।
नहीं, जब आप नीम कह रहे हैं तो आप उसमें बताएंगे कि नीम ऑयल था कि नीम कर्नल का था, कौन सा फॉर्मुलेशन आपने
हम
यूज़ किया?
हम क्या कर मैडम, हम 10,000 ppm का नीम 1 ml से हम स्प्रे लेता हूँ मैडम पॉलियर एप्लीकेशन के लिए, और ड्रेंचिंग के लिए हम 300 ppm का ले रहा हूँ। सो ड्रेंचिंग का हम 300 ppm का क्यों ले रहे हैं, वो उसका 300 का ppm रिजल्ट बहुत बढ़िया है। ओके। सो इसलिए 10,000 ppm का हम पॉलियर एप्लीकेशन कर रहा हूँ। सो 300 ppm का ड्रेंचिंग जो है ना सर, ड्रेंचिंग में बढ़िया रिजल्ट आता है। लेकिन मैं 10,000 ppm 15,000 ppm 50,000 ppm भी यूज़ किया है, लेकिन वो नियर स्टेम उधर थोड़ा हमको मिलीबग और कॉलर आर्ट का और पिन होल बोरर और स्टेम बोरर का प्रॉब्लम बहुत था। इसलिए हमने क्या किया, सो नियर स्टेम, इधर पिक्चर भी है आप देख सकता है। ये है ना, हाँ थैंक
ये पीछे करने के
यू। हाँ ये ऊपर कर रहे हैं। हाँ देखिए, ऐसा एप्लीकेशन हमने एवरी 4 मंथ्स वंस दे रहा हूँ। इसलिए हमको अभी हमारा 8 साल का पौधा है, अभी भी 1% भी मरा नहीं। पहला हम यूज़ नहीं करता, नीम यूज़ नहीं करता था। उस टाइम पे हमको 3-4 साल में हमारा पौधा मरता था वो इंसेक्ट का डैमेज के वजह से। अभी हम ये ऐसा प्रैक्टिस कर रहा हूँ ड्रेंचिंग का। 300 ppm का नीम ऑयल जो है वो 2 ml 1 लीटर पानी में मिक्स करके हम ऐसा 10 लीटर पानी 5 साल के ऊपर का जो एज का जो पौधा लेते हैं उसको 10 लीटर ड्रेंचिंग करता हूँ। सो 5 साल का बिफोर रहता है ना, उसको मैं 5 लीटर कर रहा हूँ। एवरी 4 महीने में एक बार दे रहा हूँ। सो अभी 6-8 साल तक हमारा पौधा मरता नहीं सर। अभी हमको थ्रिप्स माइट्स अभी 5 से स्टेज होता है अनार में। एक है डेवलपमेंट स्टेज, प्लांट ग्रोइंग स्टेज में, उस टाइम पे हमको थ्रिप्स का डैमेजेस बहुत रहता है। और सेकेंड स्टेज है डिपोलिनेशन स्टेज। हम क्रॉप लेता है ना सर, उस टाइम पे हम स्ट्रेस देता हूँ, पानी का स्ट्रेस। वो स्ट्रेस के टाइम पे हम छोड़ देता हूँ धूप के ऊपर। वो कितना दिन वो सूख जाता है प्लांट, उस दिन तक 40 दिन 50 डेज वो हम छोड़ देता हूँ। उसके ऊपर क्या रहता है सर, मिलीबग स्केल इंसेक्ट का डैमेजेस बहुत होता है उस टाइम पे। इसलिए हम नीम यूज़ करता हूँ मैं उस टाइम पे। स्ट्रेस कंडीशन जो होता है उस टाइम पे, स्ट्रेस कंडीशन का फोटो नहीं है इधर। उस टाइम पे हमको स्केल कंट्रोल होता है, मिलीबग कंट्रोल होता है, और स्टेम बोरर सम पिन होल बोरर वो सब कंट्रोल होता है हमको। अभी हम फ्लावरिंग स्टेज में भी नीम का इस्तेमाल करता हूँ 10,000 ppm का 1 ml पानी में 1 लीटर पानी में 1 ml मिक्स करके हम स्प्रे 3-4 बार, अभी 15 दिन में एक स्प्रे, 30 डेज में एक स्प्रे और 45 डेज में एक स्प्रे कंटिन्यूली देता रहता है सर। फ्लावरिंग स्टेज में हमको सकिंग पेस्ट का डैमेजेस बहुत रहता है, उसके वजह से हमको फ्लावर्स और फ्रूट्स डैमेज बहुत होता है। इसके कारण से नीम का रिजल्ट मेरे को अच्छा रिजल्ट मिला है। हम केमिकल भी इस्तेमाल करते हैं, लेकिन केमिकल का क्या रहता है, अभी आज स्प्रे हुआ है 5-6 दिन में रिटर्न इंसेक्ट आता है। अभी हम नीम का इस्तेमाल हुआ है 15 दिन तक हमको कंट्रोल होता है। वो उसके साथ केमिकल मिक्स करके दिए तो भी मेरे को ड्यूरेशन ज़्यादा मिलता है, 20 दिन 25 दिन मिलता है। नहीं तो एक बार हम 1 महीना स्प्रे भी नहीं देते उस टाइम पे। सेटिंग टाइम में हमको सेटिंग होने के बाद में मिलीबग का प्रॉब्लम ज़्यादा होता है। वो मिलीबग फ्लावरिंग स्टेज में मैं 3 स्प्रे लेता हूँ सर। 3 स्प्रे में हमको थ्रिप्स, माइट्स और एफिड्स ऑल सकिंग पेस्ट कंट्रोल में रहता है नीम 3 स्प्रे लेने के बाद में। जो नीम नहीं लिया, खाली हम केमिकल लिया, केमिकल में क्या होता है सर, पॉलिनेशन का इफेक्ट बहुत रहता है। पॉलिनेशन 100% नहीं होता है, वो हनी भी नहीं आता है, इसलिए हम नीम का इस्तेमाल अनार में करता है। सेटिंग अच्छी तरह से होता है नीम का। उस टाइम पे हमको थ्रिप्स माइट्स का ज़्यादा तो ज़्यादा डैमेजेस रहता है। अभी हम नीम केक यूज़ किया है 1 kg पर प्लांट सॉइल एप्लीकेशन। वो इसी कारण से हमको साइज, शाइनिंग और कलर इतना अच्छी तरह से हमको हार्वेस्टिंग हुआ है। फ्रूट का साइज और ये 2 साल का पौधा है, इसमें हमको 20 kg फर्स्ट यील्डिंग आ गई सर। उसका रेट भी 200 के आसपास हमको मिल गए। ये तो बांग्लादेश एक्सपोर्ट किया है हम लास्ट 2 years बिफोर। ये बांग्लादेश एक्सपोर्ट भी हुआ है। हम 10-15 साल से नीम जो यूज़ कर रहा हूँ, हमको अनार में इतना 50% प्रॉब्लम सॉल्व हुआ है। थैंक यू सर। वीडियो हम एक्चुअली वीडियो नहीं किया है सर, वो मोबाइल में वीडियो है, चाहिए तो अनार का।
बहुत अच्छा, वीडियो है क्या आपका इस पे?
वीडियो है लेकिन इसमें अपलोड नहीं किया ना।
अपलोड नहीं है,
अपलोड
कोई
नहीं
बात नहीं।
हाँ हाँ सर,
बहुत अच्छा प्रेजेंटेशन आपका,
थैंक यू।
जोरदार तालियां हो जाए इनके लिए।
यस यस सर। नहीं, फुल कवरेज से स्प्रे करना है सर। पहला स्प्रे करना है, स्प्रे करने के बाद में वो इंसेक्ट मरता नहीं, डैमेज फीडिंग कम कर देता है। मैं क्या करता हूँ, बाद में 3-4 दिन छोड़ के केमिकल स्प्रे मारता हूँ। 20 डेज तक हमको कंट्रोल आता है। खाली केमिकल दिया तो आपको 5-6 दिन में फिर वो डबल आता है खाने के लिए। यूज होता है, लेकिन पहले साल में मेरे को 500 ml पर लीटर ही लगता है सर।
नहीं, 500 पर लीटर की बात नहीं कह रहे हैं, वो कह रहे हैं कि एक पेड़ पर कितना पानी इस्तेमाल करते हो, कितना पानी इस्तेमाल करते हो?
स्प्रे
हाँ, स्प्रे का वॉल्यूम कितना है?
सर 500 ml होता है। 500 ml पानी 2 ml 1 लीटर पानी में मिक्स करके 1 ही ml लगता है मेरे को।
नहीं, पानी
सर 500 ml पानी पर लीटर लगता है।
आधा लीटर पानी,
आधा लीटर
पानी से एक पूरा पेड़ ड्रेंच हो जाता है।
यस आधा लीटर।
हाँ जी हाँ जी।
नहीं, हमारा क्या है सर, अभी हम यूज 30 एकर्स 20 एकर्स जो कर रहा हूँ ना, 20 एकर्स 30 एकर्स जो कर रहा हूँ ना, अभी हमारा स्प्रेयर है, हाई वॉल्यूम स्प्रेयर है। वो 800 लीटर का टैंकी रहता है। उसमें क्या करता हूँ मैं, 1 लीटर नीम ऑयल डाल देता हूँ सर।
रामू जी,
रामू
जी इधर देखिए जरा।
यस
एक चीज का पता कर दिए आपने, बताया कि नीम का बहुत सारे 3-4 चीजें बता दी आपने कि 300 ppm भी किया, 10,000 ppm भी किया, और सॉइल ड्रेंचिंग भी की, और स्प्रे भी किया, ड्रेंचिंग भी की। तो थोड़ा सा कंफ्यूजन हो गया। क्या आप बता सकते हैं स्टेप बाय स्टेप कि सबसे पहले क्या किया था, उसके बाद में क्या किया था, फिर कैसे किया था? 0 डे पे ये किया, 15 डेज पे ये किया, फिर 30 डेज पे क्या किया? एक सीक्वेंस में बता देंगे ना, अगर क्या कहानी की तरह। और इस पे केमिकल का भी बता दीजिएगा, केमिकल लगाया है तो एक कंप्लीट स्टोरी बन जाएगी। बता सकते हैं बस 5 मिनट के अंदर?
यस मैडम। हम बिफोर प्लांटेशन जो लैंड प्रिपरेशन जो करते हैं ना मैडम, उस टाइम पे 2 बाय 2 हम फिट करता हूँ।
हाँ
फिट मीन्स डिगिंग करके वो प्लांटेशन पौधा लगाता है ना मैडम,
हाँ
उसमें क्या करते हैं आधा केजी नीम केक,
हाँ
डी ऑयल केक,
हाँ।
वो मिट्टी में मिक्स करके हम पौधा को उधर प्लांटेशन हो जाता है। बाद में हम 3 दिन में एक बार पानी देता हूँ, क्लाइमेट कंडीशंस के वजह से वो फर्स्ट आफ्टर प्लांटेशन बहुत पानी देता है, वो सब एरिया वेट होना चाहिए,
हाँ
लेकिन वो रूट तो सेट होना चाहिए ना मैडम।
हाँ जी हाँ जी
हाँ, बाद में 15-20 दिन के बाद हमको न्यू शूट्स टेंडर लीव स्टार्ट होता है अनार
हाँ,
में,
हाँ।
उसमें वो थ्रिप्स का डैमेजेस बहुत होता
बिल्कुल
है।
बिल्कुल सॉफ्ट
हाँ, उस टाइम में हम क्या करता हूँ मैं मैडम, वो 15 दिन में एक बार
हाँ
10,000 ppm का नीम ऑयल 1
हाँ।
लीटर पानी में 2 ml मिक्स करके
हाँ।
हम 16 लीटर कैन जो रहता है,
हाँ।
पहला वो छोटा रहता है जब,
हाँ।
उस टाइम पे फुल कवरेज करता हूँ। 1 एकड़ को उस टाइम के हमको लगता है 50 लीटर पानी।
50 लीटर एक
यस,
एकड़
अप टू 6 मंथ,
बिल्कुल,
एवरी
up to
15
6
days,
months,
एवरी
हाँ।
15 days once।
बिल्कुल
बाय चांस, बाय मिस्टेकली वो इंसेक्ट खा लिया, उस टाइम पे क्या करता हूँ मैडम, 1 ml नीम ऑयल उसके साथ सिंथेटिक फेरिथ्राइड और एनी केमिकल्स उसके साथ मिक्स करके स्प्रे लेता हूँ। फिर हमको वो कीड़ा
ऐसा कितनी बार करना पड़ता है सिंथेटिक का यूज?
सो वो तो महीने में एक बार लगता है हमको,
महीने में एक बार।
अप टू प्लांटेशन।
अगर आप नीम नहीं लगाते तो महीने में कितनी बार सिंथेटिक यूज करते हो?
मैडम
अगर नीम नहीं यूज करते साथ में सिंथेटिक के,
यस मैडम।
तो सिंथेटिक आप कितनी बार यूज करते हैं महीने में?
मैडम वो नीम ऑयल हम यूज नहीं किया तो
हाँ।
8 दिन में एक बार जरूरी करना पड़ता है।
हाँ, तो
8
इसका
दिन
मतलब
में।
है कम से कम तीन बार तो कम से कम आपको महीने में लगता
महीने
है,
में 3 बार
जो आप केवल अभी ये एक महीने में एक बार कर रहे हैं।
यस
तो
मैडम।
2 आपने रिप्लेस कर दिए
हाँ।
नीम को यूज करने की वजह
Yes,
से। वेरी गुड, एक्सीलेंट। सो दैट्स अ वेरी गुड इंटरप्रिटेशन।
यस, थैंक यू। नहीं, वो लीव्स क्या होता है सर, सकिंग पेस्ट खाने के बाद में वो फोल्डिंग हो जाता है, सनलाइट को ओपन नहीं रहता है। सनलाइट को लीव्स ओपन है तो आपको फूड बनता है, सो फोटोसिंथेसिस एक्टिविटीज चालू हो रहता है, नहीं तो वो डैमेज होता है ना। प्लांट तो ग्रोथ नहीं रहता है, अभी 3 साल तक वो इतना ही रहता है। आप सकिंग पेस्ट कंट्रोल नहीं किया तो कोई प्लांट है, नॉट ओनली अनार, आप पपाया है, बनाना है, कुछ तो बनाना में इतना थ्रिप्स नहीं रहता है।
अच्छा, एक चीज और,
Yes,
जो
ma'am.
आपने कहा सॉइल केक आप जब प्लांटेशन करते हैं उस समय आप पिट में डालते हैं।
यस मैडम।
इसके बाद दोबारा भी डालते हैं केक को?
आओ मैडम, आओ। क्या करता है, हम डीएपी डालता है, नाइट्रोजन डालता है, पोटाश डालता है, ग्रो करने के लिए तो चाहिए
नहीं,
ना।
वो चीज ठीक है, मैंने कहा नीम केक को आप दोबारा अप्लाई करते हो?
यस मैडम, फर्स्ट बिफोर प्लांटेशन एक बार आधा केजी करता हूँ,
हाँ।
आफ्टर 6 मंथ और एक बार एप्लीकेशन करता हूँ आधा केजी, नहीं तो 750 ग्राम,
हाँ।
और आफ्टर 1 ईयर होने के बाद में 1 केजी करता हूँ।
अच्छा, एक
उसके साथ सम न्यूट्रिएंट भी ऐड करता हूँ।
हाँ।
उसका ड्यूरेशन जो है,
और ड्रिप इरिगेशन भी करते हैं साथ
ड्रिप
में?
इरिगेशन नहीं करता हूँ मैडम, लेकिन
अच्छा।
क्या है, रूट के ऊपर इतना प्रॉब्लम है, लेकिन नेमाटोड का प्रॉब्लम बहुत है। अभी हम प्रिवेंटिवली वो स्टेम के नीचे हम ड्रेंचिंग कर रहा हूँ ना, इसके वजह से हम नेमाटोड भी ज्यादा नहीं दिख रहा है।
अच्छा, कोई आपका कंट्रोल प्लॉट था जिसपे आपने कुछ भी नहीं किया?
मैडम
ऐसा कोई कंट्रोल प्लॉट था जिसपे आपने ये सब नहीं किया?
हाँ।
कोई भी नीम नहीं लाना या फिर ये जो आप बता रहे हैं नहीं किया, नॉर्मल तरीके से अपना प्लांट ग्रो किया, उसकी ईल्ड और आपकी ईल्ड में कितना फर्क आया?
20 to 30% फर्क है मैम। मेरा ये 4 प्लॉट है, एक प्लॉट को हम खाली केमिकल कर रहा हूँ, वो 3-4 साल में हमको यील्ड धीरे डाउन होके प्लांट भी मर जा रहा है, लेकिन वो उसमें इंसेक्ट का प्रॉब्लम हो।
तो आप खुश हैं? आप खुश हैं?
बहुत खुश
वेरी
है
गुड,
मैडम, इसलिए हम
एक्सीलेंट।
वो नीम के बारे में बात करने के लिए इतना दूर आया है।
नाउ एनी क्वेश्चन फ्रॉम द ऑडियंस?
मैं भी फार्मर हूँ।
यस सर।
मेरा एक क्वेश्चन है, आप फार्मर है उसी की वजह से।
यस सर।
आपकी जो वैरायटी है
यस,
वो अनार में सिंदूरी वैरायटी है या मृदुला है?
आप सिंदूरी बोलता है इधर, हमारे एरिया में बागवा बोलता है, केसर बोलता है कर्नाटक में।
आपके यहाँ जो भगवा वैरायटी बोलते हैं,
यस सर।
उसमें ब्रिक्स कितना परसेंटेज है आपके इलाके की?
ब्रिक्स के बारे में मैं नहीं पता है, मैं
मेरी
एक्चुअली
एक
वो
आपको
प्रोसेस।
रिक्वेस्ट है,
हाँ
आपने बांग्लादेश में एक्सपोर्ट किया है,
यस सर।
तो आपकी यहाँ लैब में ब्रिक्स का टेस्टिंग करवा लीजिए।
नहीं,
ब्रिक्स का टेस्टिंग करवाने के बाद
हाँ।
मैं आपको अच्छी हेल्प कर सकता हूँ।
100%
तो ब्रिक्स
सर।
के लिए तो लैब में जाने की जरूरत नहीं है, रिफ्रैक्टोमीटर आता है, वो आपको डायरेक्ट ही दे देगा।
यस 100% करता हूँ सर, 100% करता हूँ।
One
Any other question?
question? Any other question? Okay.
यस, थैंक यू सर, थैंक यू सो मच।
Thank you. Excellent presentation. Huge clap.
सॉरी, हमारा लैंग्वेज इतना अच्छा नहीं था।
It's a wonderful presentation, complete application based. Thank you very much, Mr. Tamalpo. Yeah, now I request Mr. Gurudev Singh to come to the dais to honor the speaker.
I now invite Dr. Anuradha to deliver her talk on elucidation of putative targets of azadirachtin through molecular tools from insect phenotypes to genotypes. Dr. Anuradha, please.
Good afternoon, everybody. I'm very honored to be here. This is my first participation in the conference, which is a World NEEM conference. And it's been a privilege and an honor to be part of such a conference where there are a lot of industry people and academicians who have given such wonderful talks. And for me, I have done NEEM research during my PhD times with Dr. Annadurai. And I've worked in a very small laboratory setup. So now when I'm coming out into the conference, I was very excited. But more than that, I've understood so much in just just 2 days, something that I have not pursued, and something which is happening at a mega level, and people from different industry actually coming together and in a cohort manner trying to address the issues of NEEM as a molecule to be utilized as a biopesticide in both sustainable agriculture and for medical practices. So today I'm going to be talking about the elucidation of putative targets of azadirachtin through molecular tools from insect phenotypes to genotypes. I'm supposed to sound very non-technical, so I'm trying my best efforts to convey this as much as I can. So my slides are also made in such a way which does not reflect too much of technical details, but I will try to elaborate it in a more simplified form. So we all have been going through the slides from various people about the population studies, the crop losses. I don't have to go in detail to it. People have already showed a lot of data on that. But looking at the way the population growth is happening globally, and the population growth in India, there is going to be a lot of supply-demand problems. And looking at the way things are going because of the climatic changes, a lot of infrastructure building, various other factors. There is going to be a lot dramatic that is going to be happening globally. So besides that, we also have insect crop losses, which are amounting to 20 to 40% of yield. And this is going to have an impact on the revenue loss as well. So for years together, we've known that there has been indiscriminate use of synthetic chemical pesticide. And like my previous speaker who has shown, it's very heartwarming to see that in practice, he's been able to use NEEM as a biopesticide, and he's been able to get some good yields. It's very heartwarming to know. So because we all know over a period of time, post-industrialization, there has been a rapid use of synthetic chemical pesticide, which have been very harmful to the beneficial insects and aquatic fauna. There's a lot of literature that is available, but the impact of chemicals to humans and environment has been overlooked. So this is something that we are primarily trying to address, is what I understand in this conference. And in a way, we are trying to identify if there is something, a molecule that is very insect selective. So we have some bit of data which I have generated during my PhD times, and maybe that we will be able to represent it in the current situation. So we know of particularly these vegetable crop plant pests like Spodoptera and Plutella xylostera. They are very common pests of Cruciferaceae members. And when there are a lot of chemical pesticides that have been sprayed in control of various insects. And when we looked at the literature, we found that according to the Insect Resistance Action Committee mode of action classification, in those times, there are a lot of synthetic pesticides that have been classified under various categories. But you look at this slide, you see that the primary molecule of NEEM, azadirachtin, it has not been classified at that point of time. So we didn't know the mode of action, and so we all set to understand, and we wanted to know what is the best way to go about this. So when we're talking about NEEM in sustainable agriculture, we know these are some of the factors that are very important for a biopesticide to be qualified to go into agriculture, sustainable agriculture. There is a lot of evidence of NEEM and its biological active molecules, especially azadirachtin. So now what I understood from the NEEM conference, that the biopesticide market is very small, and what the demand is quite high, although my projections may be a little varied from what has been gathered in the conference. So government aims to reduce the subsidies given to the farmers to discourage the use of these chemical pesticides. And there's been a small block study that was conducted by, I think, one of the medical research foundations. And they found that in a small block study in West Bengal, there has been farmers who have used synthetic pesticides on a daily basis for 30 days have showed cognitive decline. Although a very small population study of this kind, and they also have some neurodegenerative issues. This has been highly significant because it was a very small study, but very impactful. So can we actually help the farmers to understand that, to target insects naturally, because we always have known about NEEM, and its antefident properties, its molt inhibition properties, the delay in egg deposition, its its repellent properties and everything. And we have always attributed it to azadirachtin. Of course, there are more stories coming out, but primarily azadirachtin we've always considered as the active ingredient of NEEM, and the focus has been more on azadirachtin. So as we all know, azadirachtin has got both decalin and hydroxyfuran compounds having its own properties. One acts as a, antefidant and as an insect repellent. So whenever we are studying about biopesticide selection, we always looked at what is the invading pest species, what is the soil condition, whether there is a market availability, is there a dose selection. This is all something that we invariably walk through before identifying a biopesticide. So once a biopesticide is, a pest is exposed to a biopesticide, when it enters the gut, it ideally has to cross the barriers of the gut and move into the hemolymph and then show its action. So there are various cells, and mostly the digestion of these food macromolecules actually happen in the midgut, and that mainly may be the site of action of azadirachtin as well. So some of the scientific approach that we have used is how to address this mode of action of azadirachtin. What is the best insect model that we can use? And so we have used an in vivo model organism where we thought there is a availability of molecular tools and resources. There are a large number of transgenics that are available, and there is a screening system that we can employ, and whether we have good high-end microscopy techniques. We had collaborated with a very good national institute at that point of time, and we've also used some of the molecular tools and approaches and various immunohistochemical techniques that were available at that point of time. And we also validated it in some of the pest species like Spodoptera plutella and Helicoverpa. And what we have found very interestingly is that azadirachtin treatment induces some adult eye phenotypes in Drosophila melanogaster. What is Drosophila melanogaster? It is nothing but the fruit fly. The fruit fly that sits on bananas, and that is not very, it's not as big as a housefly to identify. It just is a very small thing. But the beauty of this insect is, although very inconspicuous, it's got a very short life cycle, roughly about 10 days. And when you can treat them with azadirachtin, basically mix them with the food and allow them to feed on it, you get a range of eye phenotypes. So at 3 ppm azav, we found that there is an array of eye phenotypes. If you see, it's a compound eye. Drosophila has a compound eye which is made up of 700 to 800 ommatidia. And they're all arranged in a very nice regular fashion. So they're basically nothing but the photoreceptor cells. And if we've noticed, some of them have even lost their eye, but they do survive. So when we looked at the other phenotypes, some of them, they could not eclose, some had deformed legs, and some even died as ferret adults. So on close observation using a scanning electron microscopy, we also found that in this disrupted ommatidial array, we've also identified there were fine bristles, hair-like structures which we call bristles, and they have a different morphology as compared to the wild type. the wild type being the Cantonese strain. So you see that there are multiple, so every ommatidia has a regular fashion of, in the corners there are bristles. But if you look at the treated ommatidia, I mean treated Drosophila, I have a disarrayed arrangement of ommatidia, and there are lots of bristles that are identified. If you see the arrows, there are a bunch of, they're not arranged in a regular fashion. So this is a very interesting observation. Now this takes us to, this is typically the life cycle of Drosophila. If you would like to know, the adult lays its egg in a day, and it goes through 3 larval instars before it gets goes into pupation, and then finally into the adult. Roughly takes about 8 to 10 days. So we use some of these immunohistochemical techniques where we've used antibodies specifically to identify the proteins of interest. For example, these are, can we move this? Yes. So this is a wild type Cantonese I imaginal disc. I'll tell you what an imaginal disc in the next slide, but just focus on this particular wild type. You have photoreceptor cells that are marked in red very specifically, and the cells which you do not see here are basically, there are cells, but they are not identified. So that is why it is very specific to the photoreceptor cells only. That's basically they are differentiating cells and not undifferentiated cells. So when we treat them with AZA, here you see that this is a magnified image, and you see that there is a line, and which we call it the morphogenetic furrow. So there is a lag in that movement of the morphogenetic furrow. So if you look at Drosophila imaginal discs, these are nothing but a batch of 60 to 70 cells which proliferate rapidly, and then all the discs, for example, the leg, eye, antenna, everything, they all go through a round of proliferation, and where there are proliferation as in they divide actively and they give rise to adult structures eventually. And this is typically how the imaginal discs are glued to the brain of the insect. Here you will have some larvae. And this is the image of, this is a graphical representation of the eye antennal disc, imaginal disc, where you have it fused, and during later stage of development, during the pupa, it gets separated. And there are lots of genes that are expressed during this time. So this is typically how a Drosophila lab looks like, where we culture them in vials, various strains, and we screen them. These are the larvae and the pupa. The fly food composition is here, and somehow I've been able to extract the imaginal disc by a simple method of turning the larvae inside out. So these are the wing imaginal discs that are treated, and you also see this is a brain, there's a control and 3 ppm, and these are stained for ecridin orange, which means the cells here are undergoing apoptosis or programmed cell death. So the cells are dying basically. And we also looked at if they are mitotically active, but we've seen that with 3 ppm azadirachtin treatment, that in the wing imaginal discs, of course it happens even in the eye disc. Here, because the imaginal discs are flat and you can really take a very good image, we found that the discs, this particular treatment, there are less number of mitotically active or the dividing cells. And this is a representation of the dying cells, which we try to overexpress P35 to rescue the phenotype of apoptosis, but that didn't happen. So coming to this cell cycle pathway in Drosophila, this is the point of action where we think that where cell division happens, that is where you have a lot of inputs coming from various signal transduction pathways, including morphogens, which are basically nothing but the diffusing proteins. And we looked at various phases like G1 to S, G2 to M, and mitotic phase, where we overexpressed some of those genes like cyclin E, cyclin A, B, and we found that there has been some rescue of phenotype. And how did we go about it? We used a very powerful system called the US GAL4 system, where you can overexpress any gene. Like if there is one copy of a gene, you can overexpress 10 copies of the same gene using this system. Now, that is fine, but how do we choose the targets? So we need to look out for some unexplored targets, which are very highly specific to insects, and that are conformationally different between the mammals and insects. So, and that is when we stumbled upon the membrane proteins and cytoskeletal proteins. The first point of contact when any pesticide encounters is the cytoskeleton. Cytoskeleton is nothing but a meshwork that defines the structure of a cell. So typically that is how the red being actins and the green being tubulins, these keep them intact. And they basically arranged in monomers and they polymerize to form the cytoskeletal networks. Now, this is a very interesting note where we found that there is a differential binding of azadirachtin on actin protein, and this differential binding, we used a bovine, it's a mammalian model where we used Drosophila model and a mammalian model, and we found that aza cannot strongly bind into the mammalian actin.
Dr. Anuradha, can
Yes.
you please wrap up?
Yes, yes.
Yeah.
This is a very interesting thing.
I
It's-
know,
yes,
but
yes, yes.
we
I
have other
understand. I
speakers
understand.
also.
Yes. So in a way, it loosely binds to mammalian actins, and that is why it does not probably have that kind of an effect as it has in the insects. And these are some of our images. We did high-end confocal microscopy, and we showed that actin is disrupted, but cyclin E, when we overexpress with in a particular domain in the in the wing, it's been able to rescue the phenotype. It's the same, and we also see that the endogenous actin, that we followed it, whether you know, that using a GFP marker, that's also a transgenic, so that also showed us the same results. In a way, we have a lot of consequences of actin disruption due to azadirachtin treatment that we all know. And we've done some mRNA expression profiles using RT-PCR in the pest species and in Drosophila as well, and we found that the muscle-specific actins have been disrupted. And this is an SDS page of G-actins, where we picked up the G-actins from the supernatant and we allowed it to run through, and we found that the F-actins are actually, there's more amount of G-actin in the supernatant. And this has been a study where we have already seen that the hemocytes are also affected, basically trying to show that actins are affected in many other cell types, including the
Dr.
hemocytes.
Anuradha, can you please wind up, please?
Yes, and yes, this is our final conclusion, that the lessons that we have learned from Drosophila is that we can use this model as a screening system because of the robustness of this system, the short life cycle, and the ease and economics of maintenance of this. And in a very small laboratory setup, we can really screen through a lot of biopesticide molecules that are not identified in terms of their targets, and also study their mode of action studies.
Thank you. Thank you, Dr. Anuradha.
Yes, yes. These are
just
my
for your,
TKs. Thank you.
for information, this is very interesting paper, but just for your information, azadirachtin is known as antiferent, okay?
Yeah.
I do not know whether you focused on that aspect also, no? Because we're known antiferent. and insect growth disruptor. So did you work on any of the target related to the insect growth regulator or antifidency?
Well, we have observed that, you know, the larvae that feed at a very high ppm, they tend to avoid, you know, but at that particular ppm where we wanted to see the mode of action, we wanted to see at the cellular level, we use that particular ppm to, you know,
Thank
do
you
our
so much.
studies.
Thank you so much. Since we are running short of time, we can have one quick pressing question from anyone if we have some quick. Yeah, please.
Yes. I'm sorry?
Any behavioral aspect, effect?
Well,
of
they may be having, but I have not followed it up. I was basically focused on looking at the proliferating cells, what is the, you know, consequence of a cell death, and, you know, the actin cytoskeletal level. But definitely there must be some effect.
Okay.
Yes.
Thank you. Thank you so much, Dr. Anuradha.
Thank you very much, ma'am.
Before we start with the next presentation, just for the benefit of the young, I see a lot of youngsters sitting here and also not so young. May I ask from the audience, who is the father of NEEM in India? Father of NEEM in India. Can anyone name how the NEEM got popularity in India? Huh? Who's the father of NEEM? That's for the phytochemistry. I'm talking about who popularized the NEEM. CM Ketkar. Anyone else? Dr. Saxena. Oh, they are all late people. First, I'm talking about the first person who actually popularized the NEEM in India. Huh? No NEEM Karoli Papa. No. I'm talking about the scientist. I think you all know him, but you are just skipping his name. His name is Dr. Pradhan. How many of you have heard the name of Dr. Pradhan? Dr. Pradhan in entomology division IRI. So you know, when there was a locust invasion in India, the entire crop was devastated. At that time, he gave the solution of the NEEM. He just made the powder of the NEEM seeds and sprayed over the locust, and locusts were controlled. That's how the NEEM became popular in India. And we have forgotten that history, and now we are reinventing the wheel. So we should know how this NEEM got popularized all over the world, not only India, but all over the world because of Dr. Pradhan. R.C. Pradhan was the entomologist in division of IRI, entomology division, IRI. You must look at the net now. Maybe those net might have archived those pages also. So we must remember the people who have contributed significantly. Dr. Govindachari, yes, for the phytochemistry, he is the one who isolated all these molecules. So Dr. Govindachari is known for the molecule isolation.
Crystallized the.
Yes,
He was
precisely.
so
he's
instrumental in
known.
crystallizing
And
that.
Dr. R.C. Saxena came very, very late. Very late.
Thank
Dr.
you,
R.C.
thank
Saxena is
very
very new.
Thank you very much.
So thank you so
I
much.
request
Thank
Mr. Gopal Sami to come to the dais to honor the speaker. Very nice lecture.
Go to the next speaker. After that high-tech research that was presented by Dr. Anuradha, now I request Mr. Ayushman Malakar to talk on 2 decades of global research on NEEM, genetic potential, ecosystem services, and pathways to sustainable development goals.
Yeah. Good afternoon, everyone, on and off the dais. I am Ayushman Malakar from ICFRE Institute of Forest Productivity, Ranchi, and today I'll be talking about how NEEM shaped the global research, particularly with a focus of genetic potential, ecosystem services, and pathways to the United Nations Sustainable Developmental Goals. My talk will be particularly a continuation of what yesterday Dr. Gurdev Singh, Dr. Tarun Kant, and today Dr. Rekha Warrior has told. So I will start my talk, my presentation, with a very ancient Sanskrit chant, Shatayur Vajradehayo Sarva Sampat Karayacha Sarvarishta Vinashaya Nimbakam Dalabhakshanam. What it translates that for longevity of 100 years, for a body as strong as lightning, for all prosperities, and for eradication of all calamities, we should eat neem leaves. This chant is actually used as the starting chant of Ugadi, which is to be held on 19th March this year. And I will not repeat these things because my previous speakers has told why NEEM has been designated the tree of 21st century by the United Nations. So in this talk, we have approached 4 research questions using a SCORBA method, that is scoping, review, and bibliometric analysis. And the 4 research questions are: what are the emerging global trends in the field of research of NEEM for the past 2 decades? What are the current knowledge base in the genetic variability of NEEM across the world? What ecosystem services can be attained for NEEM for achieving SDGs? And what are the gaps we can work upon? For this, from Web of Science database, we have accumulated 928 literatures, and through the PRISMA method of screening, we have actually worked with the 696 works. Now, from these graphs, you can see how NEEM research has evolved for the past 2 decades. Can you see the upward trend from 2018 as per global research of NEEM? And it peaked the publication in 2022 with the highest publication number of 55 across the globe. Now, if we see at the percentage documents from the last 10 years, we can see that the biopesticide research on NEEM efficacy has been surged for past decades, and it has got a very much boost after 2015 when Government of India mandated 100% NEEM coated urea. From our database, we can see that India was the highest research contributor across the globe with 230 articles, and this is also shown in the right side graph. And from India, we can see out of 230 articles, 204 articles were single country publication, that is SCP, and 26 were the MCPs, that is multiple country publications. Now, for the past 2 decades, various journals have published NEEM research, particularly the highest one, as we can all know that it was in the pest management science, the journal which publishes particularly the management side of the pests, and NEEM being a very much useful biopesticide, the research were mostly published in this journal. Now, here I present the top 4 highly cited publications from our database, and you can see that from the top 4, 2 are from India, and I am very much privileged that Dr. Nutan Kaushik ma'am and Dr. Gurdev Singh, which I have cited highly across the globe, is present here. So this graph actually represents how the NEEM research trend has evolved for the past 2 decades. From 2000 to 2008, the global research was primarily centered on NEEM's pest control, and as shown by the keywords such as pest control, NEEM, toxicity, azadirachtin A. And from 2009 to 2015, it evolved further, and during this period there was a surge. You can see that here the term phylogeny and bioassay comes because this came because for the first time in 2011, Krishnan et al. Sequenced the NEEM genome from India, and they have published that work later after refining the gene sequencing in 2016 and 2022 also. And in the third phase, you can see that 2016 to 2024 general constitutions on NEEM research has been devoted to the pest management, NEEM cake, fungicides, and as a whole its variability. So this is a co-occurrence network visualizing the key relationships in the NEEM based research, and you can see that there are several clusters, and the major cluster is particularly to the pest management and its biocontrol activities, that is shown in red. So this review was actually recently published in the TAP, Trees, Forest and People, by our team. Now this is the genetic knowledge base, and I will not delve into the details of the studies because NEEM veterans are here and they have all seen these papers, how genetic variability, how variation is present in the NEEM population across the globe. And this is the genetic constituent of NEEM, as I have earlier mentioned that Krishnan et al. Particularly sequenced the NEEM, and they estimated it to be 364 to 385 MB, the genome size of NEEM, and it actually contains relatively low repetitive DNA, which makes it a good candidate for genome editing and genetic improvement. Now historically there were 2 mega projects in world and in India, that is International NEEM Network and National NEEM Network by Novot Board, and these are already detailed by the veterans Dr. Singh and Dr. Nutan Kaushik and Dr. Tarun Kant. And I will particularly focus to the SDGs and the ecosystem services which can be gained from NEEM. Now you can see particularly we can get all four types of ecosystem services, that is provisioning services, regulating services, support and cultural services from NEEM. And when we delve into the available literatures, we found that NEEM can sustain contribute actively to the 10 UN SDGs, that is 2, 3, 6, 7, 8, 11, 12, 13 and 14 and 15. So these are much. From a single species you can get 10 SDGs. This is quite interesting, and I think that is why the UN designated the NEEM as the tree of 21st century. Now many talks have been done previously regarding the variability, azadirachtin content, oil, etc. about NEEM population, and it was evidently found that the South Indian population was very much rich in azadirachtin as well as oil content, along with other states also. But as you can see from this particular map published recently by Rajarajan et al., the eastern portion of India, particularly the states of Jharkhand, Bihar, West Bengal, along with the northeastern parts, were left alone in the exploitation of NEEM genetic resources. Here our works began with along under the leadership of Dr. Tarun Kant under AICRP 26, and this was the intervention of our institute, Indian Council of Forestry and Education, ICFRE, which took a pan-India approach under the AICRP 26 genetic improvement of NEEM, which included particularly the Eastern India, which was left along till date. And my institute, ICFRE Institute of Forest Productivity Ranchi, was entrusted with assessing the genetic variability of the NEEM population in Eastern India. So this is the map where we particularly worked. There are 3 major states, West Bengal, Jharkhand and Bihar. Along with, we took little portion, eastern portion of Uttar Pradesh, because we worked upon the agroclimatic zones 3, 4 and 7. So 3 agroclimatic zone, 3 constitutes the West Bengal state, the 4 constitutes the Bihar and eastern Uttar Pradesh, and the 7 constitutes the state of Jharkhand. Here we found, we identified 350 candidate plus trees of NEEM. What is a candidate plus tree? That is phenotypically superior than the neighboring trees in that population, and these were the selection criteria envisioned by the NOVOD board previously, and according to this selection criteria we selected 350 NEEM CPTs, and the distance between 2 neighboring CPTs were kept more than 500 meter to avoid the repetition. And as expected, we found a great level of variability in the phenotypic characters, fruit, seeds and biochemical parameters. What we assessed in biochemical parameters is oil content in our own institute, IFP Ranchi, and the azithromycin content under the leadership of Dr. Nutan Kaushik. So you can see in these pictures how much variable the seed sizes are and how much variable the oil content are. Similarly, the azithromycin content was also found to be highly variable, ranging between 200 ppm to 5000 ppm for particularly these 3 SEZs. Now, what we have found that the kernel oil content exhibited a strong positive correlation with the crown diameter of the tree and moderate positive correlation with the crown volume and GBH, girth at breast height. So what we can expect that a tree which is more thick in GBH and has a good crown diameter can yield higher oil percentage. However, it was very much surprising to find that azithromycin content showed no significant correlation with any of the parameters, phenotypic parameters we have studied of the tree. So it was actually quite shocking, but affirmative that azithromycin content is basically depending upon the edaphoclimatic factors, not so much of the physical appearance of the tree. Okay, so this was statistically validated and published also. Now, what we did, apart from this selection, we have made a robust statistical model for selection of plus trees. What is a plus tree? Plus tree is higher up than the candidate plus tree. It is proven to be phenotypically superior by several statistical methods, and for selection of plus trees we have done exploratory factor analysis which actually extracted 3 factors: seeing stoutness, tree stature and tree outspread and oil content. According to the EFA, you can find that as there was the azithromycin content was not correlated with any of the phenotypic parameters, it was left beside, it was not accounted in the variability, and we found almost 61% of variability we captured. After that we did a critic analysis. It is a actually MCDM technique, multi-criteria decision model technique, which gave weightage, number weightage to the parameters we actually record on the field, and according to this weightage we numbered the CPTs, and we identified 50 plus trees from the 3 agroclimatic zones, 10 from 3, 20 from agroclimatic zone 4 and 20 from agroclimatic zone 7. So this MCDM technique was also published in a very reputed journal, Forest Ecology and Management, by us. And after identifying the 50 plus trees, we have raised the half-sheaf progenies seedlings, particularly to be said, of 20 plus trees in our campus, and we did a multi-location preliminarily multi-location trial across the 3 agroclimatic zones from the seedlings of 20 plus trees. And the spacing was kept 3 meter by 3 meter in these multi-location trials because we were short of land. And you can see from these pictures that how from 6 months to 12 months in different areas the selected plus trees, seedlings of selected plus trees have shown the good growth. And you can see, can you believe these pictures are from Bihar, NRC Lychee, Muzaffarpur, and the right side trees are actually 12 months after the trial establishment, and those trees have gone as far as more than 4 meters in average. So these are the pictures from the multi-location trial of SZ7 that is in Ranchi. And as I said that the agroclimatic zone 4, particularly Bihar, has shown very high suitability for our NEEM plus trees, and this might be the effect of soil as well as the climate, and we are in research to find it. And this is the collar diameter chart. Now as I was telling that the edapoclimatic factor is very much edapoclimatic factor actually affects the azidectin as well as the oil content. So what we did a species distribution modeling, particularly in eastern India. What is SDM is? It is a remote sensing and GIS technique. It is a very new technique which are being applied to the forest crops. And as you can see that this SDM models the the habitat suitability of NEEM in the study area according to our what data we fed to the machine. It is effectively a machine learning process. So the 350 CPT data which we collected, we identified from the AISRB 26, we fed it, and through the RSGIS techniques we mapped out the suitable habitat distribution across the 3 states, actually 3.5 states, across the 3 agroclimatic zones, and found that West Bengal has demonstrated the highest area under the high suitable zone for this species, that is more than 21,000 kilometers square. Why RSGIS? If anyone asks, because it is not possible for us, for anyone, to survey the whole area. When we have technology to do that with remote sensing and GIS, why shouldn't we use it? And this model not only this SDM not only models for the current climatic conditions, it also predicts the future. So there are several methodologies. We have used 3 SSPs, 126, 245, 370 and 585, under which SSP 370 has shown the maximum gain. And
Mr.
as
Aishman, you have to now wind up, please.
Yeah, it will take only 30 seconds, ma'am. So by 2060, we have seen that the distribution pattern of NEEM will be projected to northward, and it is due to the climate change. So we have to think, along with the production of the NEEM, we have to also conserve these forest genetic resources. And as for the summary, I will say that from these results, true genetic gain assessment will only be possible after the plus tree progenies will reach their reproductive maturity, and then only will be the effect of genotype and environment effect will be assessed. I will complete my presentation with urging all the industry practitioners and other stakeholders to actively collaborate with us to bring these results of NEEM genetics and conservation from lab to land for a sustainable and green future. Thank you, everyone.
Thank you, Mr. Thank you so much for the nice presentation.
Thank you, ma'am.
And filling the gap of the northeastern region. So that's very nice of you.
Yeah.
I would like to share with the audience two observations which we had in the Novo Network project. There was somebody mentioned that Kerala does not have the neem tree, but it has some. There were some collections done from the Kerala, and maybe Dr. Gurudev Singh can also confirm that there was one genotype which was flowering 12 months continuously flowering. So it had flowering continuously for 12 months. That genotype, I do not know where it has gone and has not been tapped. Maybe we can again trace it back. Dr. Gurudev Singh, would you like to comment on that? And then second thing was that I'm very happy to see that the impact of the Novode project was that there was a target of the plantation of the neem trees in that project, not only the collection characterization, but the plantation for the CPTs which were identified was also there. And there was everybody, because institutions were not ready to give that much of land for the plantations. So what the institution had done, they did the plantation across the highways. And they got so much of scolding from the chairperson of the task force that why have you done this like plantation on the roadside? Nobody will take care of them and they will be not seen after 5 years. But I'm happy to note here, to state here that when I go to this Noida, Greater Noida Expressway, when I see the neem trees, I'm very happy to see they all are very healthily growing. And that's the outcome of that Novode Network project. So all the neem trees which you see across the highways is mostly the plantations done under the Novode project. So and that's very good contribution apart from this characterization and all that. So I just wanted to acknowledge that. Thank you so much. Now we can have one or two quick questions for Ayushman. Yeah, please, sir.
Yes, sir.
As a content, neem seed in Bihar.
Okay, sir, for the azadirachtin content for Bihar, it was quite variable, and it was, as far as I remember, it was variable, ranged between 400 ppm to 2600 ppm. Not very much. We were primarily focused to the oil content, and as you had all seen that out of the all agroclimatic zones, SEZ7 was one of the highest performers in terms of oil content, and we are particularly focused on the genetic improvement of neem, and as always said that selection of good mother tree is the primary, should be the primary focus of any industry or breeding activities, because without the good mother tree selection you cannot get good yield. So
anyone?
yeah. Okay, I will answer this in short, as I have shown you that from the scientific study also, before EICP 26, Eastern India and Northern Eastern India was totally secluded. There were, I think, very few 10 or 20 collection in the Novode board from this area. So this is a totally unorganized sector till now, the collection, neem collection in the Eastern India and the Northeastern India. So there is no such like collective, farmer collective.
You
who
didn't
are-
do the ex-situ conservation of these germplasm? Ex-situ conservation?
Yeah, ma'am.
You
Except
have
we
in your institute? or-
Yeah, yeah, yes, ma'am. We have the institute as well as in three SEZs.
In all the three SEZ?
Yeah, yeah, all three SEZs we have that
So
ex situ
ex-situ conservation
plantation.
इन्होंने अपने उसी को मल्टीप्लाई करके
हाँ.
अपने यहाँ लगाया हुआ है उस चीज को.
Yeah, and we are in the process to tell the farmers how to
Okay.
that.
Finish.
So
Yeah, yeah.
yeah. Huh. Yeah.
Yes, we have done the fatty acid composition. 100%, not for the nimbin and selenin, but for fatty acid composition. Yes, 100% profiling. 100% profiling for the fatty acid composition has been done under the NOVOD project. Oil content, yes, yes. NOVOD oil content and fatty acid composition, both were done. All chemical composition. Yes, yes, all that grading. 100% mapping. I had those oil samples like stored with me for around 20 years. After that, I discarded them.
Yeah.
Yes, it is there. Yes, yes. The oil was 100% profiled. Yeah.
Sir,
All the collections were profiled.
Yes, thank you. Sir, I will just urge everyone that while the industry, veterans and the NEEM scientists are looking towards the chemical constituents production of the NEEM, I urge everyone to also look into the genetic resource of NEEM, because if we lose these genetic resources, there will be nothing.
And one more thing, it's not only seed, the leaves are our
Yeah.
pharmacy.
Yeah.
We are not looking at the pharmacy of the world. These are the leaves. And this is wonderful because घर का जोगी जोगना आन गाँव का सिद्ध वाली बात है. So I think we should look at the leaves also, focus on leaves also, do some network research on neem leaves.
Yeah, thank
So
you
that
very
is
much.
very much required.
Thank you very much, Doctor. Now I request Dr. Susie Reynolds to come to the stage to honor the speaker.
Thank you, everyone. Please put your hands together for Dr. Nutan Kaushik. Now this sapling will be found on another highway in few years. Now I would like to invite Mr. Milan Mehta. He has an announcement to make.
Yeah, she announced. So everybody here, just to tell you, NEEM has a endorsement program where company and WNO endorsement is there on the product, if you have seen the endorsement. So as a part of our endorsement, we are promoting that product also here now on the stage, and I would request everybody to collect this free sample till the stocks last from the stall. Please do so. And I would also request all other members who are into B2C product, please contact WNO Secretariat for this endorsement program, which is going to promote your product worldwide. Thank you very much.
Thank you, Mr. Milan Mehta. So Nimail is up for grabs, and I hope that one day WNO becomes very much like the Silk Mark or Amul. We are having great hopes. Now I would like to invite you all to second floor for lunch, and we will gather back here again at 2:30. I know that I'm giving a very short time for you to enjoy your delicious food, but some of us have flights to catch and nobody would like to miss the upcoming lectures. We have a few lectures including the NEEM project by Manasvi Sridhar. Thank you. Let's meet here back at 2:30.